Hey young biologists, by now you should have completed this card sort, and hopefully what you got is right here. So if you need a second to double check your work, go ahead and pause this video and make sure that yours ended up in this order. Now, this part, it's really important to be able to sort through and figure out what's going on in these pictures. That's our skill A for this class. Representations and models. And in this case, these cards are models and representations of DNA replication, the process where DNA copies itself. Now, DNA replication has some steps that I want to make sure that we write down on here, and DNA replication uses some enzymes. So let's write down those steps and talk about the enzymes that are involved in each process. So the reason this one is the first step is because you can see that the DNA has the original strands here. The first step shown right here is that DNA is unwound and unzipped. So the DNA goes from being wound in a double helix to being unwound, and there's some unwinder enzymes here. And then DNA goes from being just unwound to being both unwound and unzipped, and it says unzipper enzymes. Now the truth of the matter is that those enzymes have a name, and that name is actually the same. So DNA is unwound and unzipped by DNA helicase. I really like the name of that enzyme because it gives me a clue of the job of that enzyme. DNA helicase unwinds and unzips the DNA double helix. So these unwinder enzymes and these unzipper enzymes are DNA helicase. Now, once DNA is unwound and unzipped, now some new DNA is going to start to get built. And that's why these builder enzymes come into play here. And you can see that the builder enzymes are at work in this fourth step and in this fifth step. At the fourth step, the builder enzymes are just getting started with their building job, and at the fifth step, you can see that the builder enzymes are almost done. So, our next step bridges between 4 and 5. Here's step 2. DNA polymerase builds new DNA, and I'm going to continue on over here using old DNA as a template. So what that means is DNA polymerase, not only is it a building enzyme, but it's kind of a reading enzyme too. DNA polymerase reads the old DNA molecule, and wherever there's an A, DNA polymerase will put a T to match up. Wherever there's a C, DNA polymerase will put a G, and so on. So these enzymes, builder enzymes right here, 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 and here, I'm going to go ahead and write their name because I think we could handle that. Up here, I'm just going to draw an arrow, and down here, I'll write the abbreviation, DNA Pol for DNA polymerase. By step 6 here, the new DNA has been built using the old DNA as a template. So now we've got to do something else. It's really important that when DNA is copied, the DNA is copied exactly the same and no mistakes are made. Mistakes in DNA are called mutations and can have disastrous results for cells. DNA mutations can cause disease, can cause cancer, or can even kill the cell. So if there's mistakes, we want to catch them before this process is done. And that's the beauty of having these spell checker enzymes. Now, we're just going to call these enzymes spell checker enzymes. We're not going to use the fancy name here. So, step 3 is that spell checker enzymes find and correct any mutations. Once those spell checker enzymes have done their job, DNA replication is done. At the end of this process, what we have is 2 new double-stranded DNA molecules. And just to show you, I'm going to show the old DNA molecules. I'm just going to circle them with my brown pen. These are the old ones. And the new DNA molecules, I'm going to circle those with my blue pen. So the DNA polymerase builders build the blue molecules using the brown molecules as a template. And this original strand, those are both browns. All right, that's this process. Hopefully, looking at it with this model gives you a different spin on things, and you feel more comfortable in Skill A for DNA replication using models and representations to understand this process.